So now that we know the structure of motor units and how the nervous system is connected in a way to stimulate muscle fibers, how does this stimulation actually occur? Well, where it really happens is known as the neuromuscular junction. The neuromuscular junction is where that nerve fiber meets with the muscle fiber or the muscle cell. And this is where that innervation of the skeletal muscle occurs. And we're going to introduce several new terms. These terms will be used throughout describing the nervous system. This connection or this place of the neuromuscular junction is called a synapse. A synapse is a space between, or an area between, where a nerve fiber meets its target cell. In the nervous system, a synapse is going to be between two neurons, two nerve cells. In the neuromuscular junction, it's where that nerve fiber meets its muscle cell. When the target cell is a muscle fiber, the synapse is called that neuromuscular junction or motor end plate. The axon terminal is going to be the end part, as in terminal, of the axon. If you recall from our neuron shapes, they can have crazy shapes, but essentially they've got a cell body with dendrites off of them. They have these long axons where they send messages down, and then they have an axon terminal. And this terminal is where it's going to meet the muscle fiber. Okay, so this is what we're talking about is the terminal. It's a swollen end of a nerve fiber. It contains something called synaptic vesicles. Well, we learned in our cell chapter, vesicles carry things across the cell and can carry stuff to be exocytosed and released, and that's what these synaptic vesicles do. They carry something called a neurotransmitter, and in the neuromuscular junction, that neurotransmitter is specifically called acetylcholine. So these vesicles are going to carry acetylcholine that were made in the neuron, and they're going to exocytose them into the synapse. That's why they are synaptic vesicles. They're vesicles carrying something meant for the synapse. A nerve impulse that originates here in the cell body is going to travel down the axon. It's going to depolarize the membrane, making it more positive, and it's going to cause those synaptic vesicles to fuse with the axon terminal membrane and exocytose that acetylcholine into the synapse. Now, the acetylcholine, as we can see here in this figure, these little green dots are the synaptic vesicles with acetylcholine. The acetylcholine, once released, will go across that synapse space, and we call that the synaptic cleft. So it's that space or narrow gap between the axon terminal and the sarcolemma of the muscle fiber. So the acetylcholine will cross that synapse. First, let's take each of these parts of the synapse in turn and just review what happens at each. Here we have on the left an electron micrograph of a synapse with a postsynaptic membrane. When we use terms like presynaptic, postsynaptic, we're talking in relationship to the synapse. So remember, this neuromuscular junction is a synapse where you have a motor neuron, which is here, or a motor nerve, and it's meeting with a muscle fiber or a muscle cell. We call this side the presynaptic side because it's pre the synapse, and we call this muscle cell the postsynaptic side because it's on the after part of the synapse. So this action potential arrives down the axon, gets to the axon terminal, and it depolarizes. What happens in depolarization is it's becoming more positive. So the membrane potential of this axon terminal will become more positive. And what this does is this causes calcium to come in, and the calcium is needed to cause exocytosis of those vesicles. And that will cause the acetylcholine to be exocytosed into the synaptic cleft. the acetylcholine diffuses across the cleft, and to prevent acetylcholine from constantly being in the cleft, we have something called acetylcholinesterase. Acetylcholinesterase is an enzyme that actually breaks down acetylcholine, and this will keep that muscle cell from constantly being stimulated. It allows us for relaxation of the muscles. So then we move, as that acetylcholine goes across the cleft, then we've moved to the postsynaptic side. And on the postsynaptic side, you've got the sarcolemma, which, remember, sarcolemma is the fancy muscle name for the muscle membrane, muscle fiber membrane. And what the sarcolemma has is a lot of acetylcholine receptors. These are ligand-gated receptors that will bind acetylcholine. So if you remember when we were talking about the different types of transporters on a cell membrane, this is one type. They're receptors that are going to bind a ligand. And in this case, our ligand is acetylcholine. The postsynaptic membrane then has these acetylcholine receptors, and they have folds as well, which you can see in this electron micrograph. And by having these folds in the membrane, it allows it to have a lot more acetylcholine receptors. So there's a lot of these here that can bind our acetylcholine. When the acetylcholine binds, it opens up these receptor channels, and the channels allow sodium to come in. And so sodium carries with it a positive charge, and that positive charge will cause a depolarization event. When something becomes more positive, it is depolarized. And this depolarization will then be transferred down the sarcolemma. And eventually that charge will reach the sarcoplasmic reticulum. When there's not enough receptors, you can get weakness in individual muscles, and this is the disease known as myasthenia gravis. The NMJ also is enclosed in a basal lamina. The purpose of having this enclosure in a basal lamina really helps keep the acetylcholine localized to the synapse, where it's not just floating everywhere, and it keeps this relationship pretty intact. It helps with injury, to repair injury if the synapse ever gets injured.